job. So as you all already know, he is a candidate for our ecology faculty position, which is replace the Dr. Carl Stepton's old job. So yeah, Ian is I would say the local product, okay, and get a bachelor degree from here and the same building, and we were every day. So he get a bachelor from UALR, and then cross river and get a master at UCA, all in the ecology, environmental science, organismal biology area. Then got his PhD in Ohio, very northwest end of Ohio, right at the corner with Indiana. So then he moves on to do a postdoc research all the way to south to Miami. So it's like from
About as far as you can go.
yeah, as far as you can go. So it's still working on ecology kind of stuff, field study, as you can see from here. So is still very early in his career, have only 1 year-ish, 1+ year postdoc, but already did demonstrated his great in research, and he can teach this in the biology course, and he published paper as a master student, as a PhD student, and start put the manuscripts together from his postdoc research. He's a very productive research young scientist. As a student, and he earned a lot of small fellowship, travel award, meeting award for his research from a master's program and PhD program. So he's a very good role model for all students, for all junior faculty here. So today, and we have him here, and it's late, and it's in summer, so I apologize. We don't have a lot of audience here. So as I mentioned, all the important people are here, and so just you have like 45 minutes to talk about your research, and if people have questions, then we have a few minutes, 5-10 minutes question
Sounds good.
session. Okay? All right, great. That's all yours.
Well, thank you guys for coming. Yeah, I totally understand with the summer. It's pretty empty on campus. So my name's Ian Clifton. I'm currently a postdoc at Florida International University. And I'm, just real quick, this is Utah. This is in Canyonlands area. So this is where I did some of my dissertation work. So I am a physiological and evolutionary ecologist. I'm really interested in the processes and interactions between ecology and evolution. And this typically has a human-mediated context to it. So looking at things like responses to land use changes, climate change, things like that. And I usually use a combination of lab and field experiments to address my questions. So a lot of things like environmental manipulations, like transplant experiments or common garden studies. So just real quick, we know that Earth is currently going through this period of extremely rapid environmental change. This is things ranging from changes in land use, like that causes habitat fragmentation, increasing urbanization that's increasing interactions and conflicts with natural environments, and of course climate change, which is shifting these, causing things like massive increases in global mean temperatures, increasing climatic variability, things like that. Now, in the face of such extreme and rapid change, many organisms, particularly the ones that are less mobile, are essentially forced into one of two paths. They either change or they risk going extinct. So this extinction example here is a projection for high elevation lizards under the lowest climate change scenario. And we still expect quite a few to be extirpated and potentially extinct. So when we think about organismal change, one of the primary models that we think of is this adaptive response. That is, organisms experience pressure from the environment. That pressure acts on phenotypes, the physical expression of the underlying genotype. And organisms that are more suited to the environment are more successful, they live longer, they produce more offspring, they contribute more genetic material to subsequent generations, and we see this phenotypic shift that matches the environment, and that is associated with an underlying genetic shift, right? But of course, like most things in biology, it's rarely that simple. My main interest for me is phenotypic plasticity. So this is the ability of a single genotype to express multiple phenotypes depending on the environment. So another way of saying that is the phenotype and the genotype are not one-to-one. So just because you have pressure on a phenotype does not mean you'll necessarily see changes in the underlying genotype, right? And this is important because adaptation occurs over a relatively long period of time, takes generations to occur, where phenotypic plasticity occurs within a generation, within a single individual. And that individual may change, and that individual may persist, but they may not pass any of that onto subsequent generations. I'm also interested in this idea that phenotypic plasticity and adaptation can interact, and we have two main hypotheses for this. The first being facilitation. So that's this idea that phenotypic plasticity, which is a really fast mechanism, can move an organism's phenotype close to a phenotypic optimum. So if we think about this on a fitness landscape, it moves the phenotype towards a fitness landscape. That gives evolution, adaptive change time to catch up, and you slowly move towards the peak of that adaptive landscape. Alternatively, we have this idea that phenotypic plasticity may actually constrain evolutionary change, constrain adaptive change. This is really an important idea in my field of thermal biology because one of the main areas that we see this occurring is through behavioral modulation, behavioral plasticity, and that is that organisms that are in an environment where they can't behaviorally thermoregulate are exposed to selection. But organisms that are in an environment that is really heterogeneous and they can avoid critically high temperatures rarely actually meet these critically high temperatures, and that shields the underlying genotype from selection. And that's important because it can be useful in the short term, right? But then as climate change outpaces this behavioral ability, the underlying genotype has not shifted with it. So in my research, I am essentially trying to address this one overarching question: how does plasticity mediate responses to environmental change? That's plasticity itself, as well as its interaction with adaptation. And I'll say that this was not necessarily my plan going into research. I've always been really interested in adaptation. But a lot of what I've done has kind of shown how important plasticity is for mediating these responses to environmental change. So today I'm going to talk about my research in two systems. I'll start with morphological and prey variation. That's how prey variation affects predator morphology. Then I will talk about climatic and physiological variation, so how climate acts on the physiology of organisms. And then I'll spend just a few minutes at the end talking about what I would like to do here, kind of what my priorities are if I were to take this position, get this position. So jumping into this morphological and prey variation, we know that landscape modification imposes new pressures on organisms. If we think about that in a predator-prey context, we have things like land use changes for agriculture that can increase this edge space. It can cause population declines in important prey species that can affect predator abundance and things like that. You also have, again, more landscape use that can just cause all sorts of effects on the prey communities. One example, or a few examples of this, are the introduction of round gobies to the Great Lakes region. So round gobies are this invasive little fish that have spread throughout the region, and while they caused a lot of harm to the local fish community, they actually helped rebound the snake community that was eating them. So the prey population, or the predator population, had declined, and this new food source comes in, and they're actually able to rebound. We also have this really classic example of the trophic morphology of soapberry bugs matching introduced soapberry plants. So they were these shifts in proboscis length related to the depth of the pit that these bugs were trying to reach. So here, this is work that I did in Arkansas. This is in Roanoke County. This is the Arkansas River Delta region. It's very well known for its agricultural output, rice, soybeans, all that kind of stuff. One thing that's maybe less known, particularly to people outside the state, is that it is also a huge aquaculture region. So anyone that's ever been to maybe UAPB and checked out their facilities knows that Arkansas and aquaculture kind of go hand in hand. But this region is dotted with all of these fish farms where they raise fish for all sorts of commercial uses. And this is really interesting to me as a researcher because these farms actually specialize in the types of fish that they raise. So in my language, they specialize in the types of prey that they raise, which they don't appreciate when I call them prey. So some of them raise these large-bodied fish, things that you use for food, things that you use for game fish, to stock lakes and ponds. But ultimately, these just really large fish. Others specialize in raising these really small-bodied fish, so things like goldfish and fathead minnows. So what that's effectively done is created this semi-natural mesocosm system in the area, where you have these expanses of agricultural region and these dotted aquaculture farms where these snakes can inhabit. And I skipped ahead a little bit, but so I'm interested in snakes as the predator. Snakes are really common on these farms. This is the diamondback water snake. These are primarily fish eaters, and while that may seem obvious because they're water snakes, most water snakes actually eat a whole lot of amphibians. These guys don't. They almost exclusively eat fish throughout their entire life. So they're kind of an ideal model for this system. And if you go out on the right nights in the summer, you can find dozens of these snakes pretty easily. So what we did is we would go out and we would capture snakes at these 4 fish farms. 2 specialized in raising small-bodied fish, 2 specialized in raising large-bodied fish. And we were interested in this idea of whether or not prey size affects predator trophic morphology. Right? So trophic morphology is a common response. That's the feeding apparatus. Is a common response in predator morphology to prey types. Think back to the soapberry bugs and their proboscis length. And snakes are kind of perfect for this because all it really is a head attached to a tube. So there's not much for selection to act on, and there aren't many responses that can occur. So we wanted to know if we saw an increase in body size that was related to the size of prey that these snakes are consuming. One important thing I forgot to mention, but I want to make clear, is that there is a distribution of prey sizes at these farms. It's just highly constrained. So it's not like if you go to a large prey fish farm, you're only finding fish this big. Obviously they're raising some fry and stuff, but they're all much larger than what you'd find on the small prey sites. And what we see is that there is this concomitant shift in predator morphology related to prey size, where snakes from the large prey populations were reaching larger maximum body sizes than snakes from the small prey sites. But interestingly, we only see that in females. And this is potentially because there are multiple selective agents acting on females that aren't acting on males. And that is primarily that bigger female snakes have more offspring. So the more offspring you can produce, the more genetic material you add to the next generation. So it is directly related to fitness. So that begs the question, skipping a little bit, but again, just reiterate, larger body sizes in females. So that begs the question, what processes drive morphological divergence, right? You can have local adaptation where selection selects for a larger body size through some mechanism, Or we can have a plastic response where because you're eating bigger things, you get bigger. And that could be a consumption response, obviously, but it could also be a change in energetic pathways where energy is differentially allocated to somatic growth versus other things. And we would expect to see, if this is an adaptive response, that growth rates would be similar between populations, but their mean would be different. Where if it's a plastic response, we'd expect to see this change in growth rate depending on the size of prey that they're eating. So the way that we addressed this was we focused on just 2 of our original fish farms, one raising small, one raising large fish, and we would go out and we would collect pregnant females, and pregnant is the right word here because these are live bearing snakes. We would bring them back, let them give birth, and then we would take 2 females from each litter and we'd split them into different feeding treatments. They would either be fed large prey items or small prey items. So this figure on the left here is just showing that we fed the large prey treatment, much larger prey items, and maintain that as well as increasing it, while we kept the size of prey for the small prey treatment the same over the course of the study. And what we end up seeing is that, first off, the snakes from the large prey population that were consuming larger prey items actually did have a steeper growth rate. And this is corrected for consumption. So they were consuming relatively similar amounts of food, yet they were growing faster. So that is at least some evidence that this is a plastic response to the size of prey itself and not necessarily just increased consumption. The other thing I'll note is the growth rate is not different for snakes from the small prey population. There's a lot of variation there, particularly towards the end. And there's an interesting explanation for that I'll get to here, a potential explanation for that I'll get to here in a second. So what we see is that there is some evidence that plasticity is contributing to this divergence that we see at the adult level, right? So if these snakes are consuming larger prey items, they're having faster growth rates, they're getting larger at these large prey sites. We also have this evidence that larger offspring may be more successful. So if you just look back at the conceptual diagram I show you here, this local adaptation would be a shift in means. And we do seem to have evidence of a shift in means, right? So we see that these snakes are born larger from the large population, and that difference is maintained throughout the course of the study. And one potential explanation for this is if we look at the consumption, so how we were feeding these and what snakes in the small prey population were eating, we see this interaction. So while they were eating the same amount over the entire course of the study, that shifted partway through. And what that may indicate is that the snakes that we were feeding large prey items to from the small prey population were actually too small at birth to consume the prey items. So one way of looking at this is, well, we kind of messed up and we should have fed them such large prey items. But it's also evidence that being born larger is an advantage if larger items are more prevalent. And actually where we see this shift occur and they start consuming similar rates is at the starting size of snakes from the large prey population. So we also have evidence that this is an adaptive response, right? And that's maybe not as fulfilling as we would like. We can't say it's definitely this or definitely that. But we have this idea that these two have interacted. And so what we can learn from this study is, first, that morphological divergence can occur really rapidly. So the oldest population here has only been around for about 70 snake generations. So that's a snake generation is under 2 years. So these shifts are really rapid. And that may be evidence of this facilitation effect that I mentioned, where plasticity is moving them to the new local optimum on these large prey sites, and then over time, adaptation moves them closer. And importantly, that large prey having larger offspring, we can't rule out the idea that that's transgenerational plasticity or something like that, maternal effects. So there are a lot of really, I think, interesting questions left to answer in this system. And on that, I'll just talk about like some of the questions that directly come to mind, some of the things that I would like to do in this system were to come back. First, do these patterns exist in a broader metapopulation context? So if you look at the waterways and the pathways that these animals could move through on the landscape, you can see that there are actually a bunch of like what look like probably fish farms and almost certainly are fish farms, as well as some natural areas. So looking at whether or not these patterns exist and trying to figure out some way to potentially quantify the connectedness of these pathways. I think one interesting project potentially for someone like a master's student is, what are the functional limitations on prey size? This is a really important question for snakes and other gape-limited predators, but how big is too big, and what changes have to occur, not just within the body, but within the head itself. So snake skulls are exceptionally kinetic, and independent bones can shift relatively easy. They're relatively evolutionarily labile. And then I'm going to talk more about this at the end, but one of the things that I would really like to do is look at the actual mechanisms that underlie changes in growth rate, plasticity and growth rate. So using things like transcriptomics to look at what mechanisms are, what genetic mechanisms may be underlying this. So next I'm going to kind of shift gears and talk about climatic and physiological variation in lizards. This is a greater shorthorn lizard. She is very pregnant because she's also a live bear. So you may have noticed that I tend to focus on reptiles. So real quick, one of the reasons I do this, particularly for thermal biology, is that one, reptiles are thought to be quite vulnerable to climate change. And this is because reptiles are cold-blooded, they're ectotherms, right? So what that means practically, and how we describe it as thermal biologists, is with what's called a thermal performance curve. This describes the performance of some metric. Broadly, we say physiological performance, but metabolic rate, locomotor performance, just some ecologically relevant trait over a range of body temperatures. And I'll point out some of the most important features for what we're talking about, but what you should notice is that the curve increases gently with increasing temperature until it reaches this point, after which it just drops precipitously. And that's thought to be a biochemical response where proteins start to denature and physiological function is just gonna tank anyway. So the most important things for us to think about for the context of the studies that I'm about to talk about are the thermal performance breadth. That's the entire range of temperatures an organism can withstand while maintaining some level of physiological function. And it's bracketed at the high end by the critical thermal maximum, the upper thermal tolerance limit, and the low end by the critical thermal minimum, the low or the lower thermal tolerance limit. So thinking about this more broadly, thermal physiology is really interesting because we have these expected trends in how thermal physiology should vary across environmental climates. One of the biggest ones that people are interested in because of the due to the consequences of climate change is this global trend where organisms in more aseasonal environments like the tropics should have a narrower thermal tolerance range because they've constantly evolved in really stable climate, right? So they're not experiencing a lot of stochasticity. On the other hand, in more seasonal environments, we would expect organisms to have a wider thermal tolerance breadth because they have this really seasonal, this really stochastic environment where they've evolved. But even within that, we have these ideas of how seasonality may affect thermal physiology, right? So one thing that we might expect to see, and in general we expect, is that seasonal environments should select for more plastic organisms. So like I said, that interaction, plasticity itself can be adaptive. So in a seasonal environment, we might expect when it's really cold, not getting very warm during the day, when temperatures are basically more constrained, the thermal tolerance breadth may also be constrained if the animal is physiologically labile. compared to middle when it's still getting cool at night, but it's getting very hot during the day. So this is all work that I did in San Juan County, Utah. This is just outside Canyonlands National Park. And this area is it's on the Colorado Plateau, so it's considered a high desert. This is over 1,500 meters in elevation. I'll try to call out the elevations somewhat. But so this is a high desert, and these places are known for being incredibly variable, right? So from day to day, you can have huge swings in the weather and the temperature. And this was actually, I showed up to my field site on May 18th, and there was snow on the ground, which, if you know anything about lizards, is not prime lizard habitat. But they were out and about the next day on May 19th. The importance of a system like this is that the Southwest is actually expected to change more than any other region of the U.S. This is part of the reason that we're seeing such a huge increase in the fires that are going on out there, these really long-standing droughts. So understanding how organisms in this region are dealing with climatic variability in general is really important for understanding how they can respond to climate change. So here I focus on two species of Phrynosomatid lizard. This is the plateau fence lizard. They look very similar to anyone that's looked outside and seen a fence lizard because they are pretty closely related. And then this is the common sidewatch lizard. This is a really common experimental animal because they are super, super common out there and they're pretty easy to catch. And going into this, we were interested in understanding how these animals deal with climatic variability. And we were addressed this by going out, sampling animals, measuring these thermal tolerance limits at different points throughout the years. So in 2017, we measured these animals in August. So in August, it's really hot, and it still gets pretty cool at night because it's still, like I said, a high desert. And then in 2018 and 2019, we went out pretty early in the year. So in May, when it's actually really cold at night, and getting warm in the day, but not necessarily peak temperatures. So what we see is that there is this constraint in this air temperature range. Notice this figure is the range of air temperatures. So if it's lower, there is a lower variability, and if it's higher, there's higher variability in air temperatures. And what we see is that thermal physiology actually matches these air temperature trends. So when we went out and checked these animals in 2017, we found that they had a narrower thermal tolerance breadth compared to 2018 when we went earlier and find that they have a broader thermal tolerance breadth. That is, they can tolerate more temperatures when you're out there earlier. So we were interested in understanding how that can occur and where those changes might happen, right? Because the thermal tolerance breadth is just that range of temperatures, but we would expect to see the changes at the high end or the low end, or maybe both. But in fact, what we see is that CT max is highly stable. Doesn't matter when you go out, CT max tends to be relatively fixed for both species. And this is important because this is actually something we would expect to occur, because CT max is the thing that's easiest for them to avoid, right? These lizards are out there. If it gets real hot, they can just go under a rock. They can go into a burrow. But if it's getting really cold, you can only go so deep before you are meeting those minimum temperatures. And indeed, what we see is that CT min is ecologically labile, as we would expect. So if you go there earlier when it's getting colder at night, they can tolerate colder temperatures. And just looking at this another way, we can see that if we compare minimum air temperatures to CT min, there is a positive relationship, and minimum air temperature to thermal tolerance breadth, there's a negative relationship. So that is, as minimum air temperature increases, CT min also increases. So some takeaways from this study: stochastic environments do seem to select for more physiologically labile organisms. That is, organisms that are more plastic. Seasonal plasticity likely minimizes costs associated with physiological mechanisms. It's a little less clear on the low end what these costs may be, But you have to think about when organisms are, releasing, say, cellular metabolites that are helping avoid freezing, as some frogs and turtles do. That producing that kind of stuff can be costly, and turning that machinery on and off can be costly. So lability helps to avoid those costs while maintaining the benefits. And this also suggests that these desert species may be intolerant because of that constraining of behavioral plasticity. Because CT max is rarely met in the field currently, CT max is evolving. But as we experience this greater climate change, these animals may not be able to keep up. Once those habitats become unsuitable, they won't be able to continue to behaviorally thermoregulate. So with that, I'm going to talk more directly about climate change in a project more explicitly investigating climate change effects. So like I said, we're experiencing this period of warming. The Southwest US is going to be likely impacted more. And this also has increases in climatic variability. And one of the things that we often think about in this context is heat waves, right? So how do animals deal with these infrequent but really intense heat waves? We know that climate change can cause performance declines in animals. So this is a study we did in 2020 where we exposed lizards to high temperatures for 3 hours, and we see that after that they're not able to run as fast. And then this is another study that shows that climate change reduces habitat suitability. So that is, as temperatures warm, habitats where these organisms could potentially thermoregulate become unsuitable. And I'll just note, this is not our study, this is in the tropics. Behavioral plasticity is really important because it can help mediate those effects, right? Like I said, if it's really hot, you can often just go under a burrow, go into the shade, just like we would do. And that helps these animals maintain this peak physiological function. But that in itself can be costly, because if you're having to move a whole lot, then you're having to expend energy to move. So this is a study that was actually done here at UALR before I joined the department, but this is looking at collared lizards, and they find that they sacrifice growth rate to modulate their behavior. So if you have alternating light sources and they want to heat up, they're having to run back and forth and they don't grow as fast. So one of the reasons that I'm focused on this high desert ecosystem and that it's important is because we know a lot about tropical lizards, actually, because everybody, all the folks working in this field have recognized that tropical regions are going to change really fast. But less recognized is that desert regions are also going to change really fast for very similar reasons to the tropics. That is, this increased stochasticity is going to be felt more acutely in this region. So this is a study that occurs higher up in elevation, so it's the same general area, but we go out of the canyons. The species we work with is a high elevation specialist, and in this area it occurs between 2,000 meters and 2,500 meters in elevation. Kind of jumped the gun on the slide. But this study that I kind of mentioned briefly, where they're predicting declines in high elevation species, these are actually, many of these regions correspond to high elevation deserts. So this is very likely to be an acute problem pretty soon. So one of the first projects I worked on during my PhD and going into this system was looking at how these animals deal with acute climate change, acute temperature change. And what we see is that these animals can very rapidly alter their behavior. So this is a reciprocal transplant experiment where we took lizards from a high, cool elevation and moved them to a low and vice versa, and we see that they match the behavior of the local lizards. And this is actually really interesting because this is using some cool technology that's typically used for bird migration to look at basket behavior. So the species that we're using here are the greater shorthorned lizards. This female is exceptionally pregnant, and she's soaking up the sun. And you can actually, I don't know how well it shows up because I did a great job painting, but we have this backpack on here so we can follow her around, and we can also measure the light that she's experiencing. So this animal, their main form of defense is running away and trying to hide. And there's a lizard in this picture, and I took the picture because I just set the lizard down and still couldn't find it. I did a less good job of painting their backpack in this one. But I'm ashamed to say I've actually lost several lizards because of this. So for this study, we did a one-way transplant, right? Because climate change, while it is going to have some effects on the cold end, the most acute effects are going to be on the high end. So this is one way to think of this context is a heat wave, right? So what we did was we took lizards from high elevation and we moved them down in elevation for 2 weeks to an area that was about 3 degrees warmer. So this is the high end of their elevational range at 25-ish hundred meters, and this is the low end at 2,000 meters. And we also have our control group that we tracked on the mountain to see how they responded to this as well. And the first thing we wanted to do was make sure that we saw that same sort of behavioral shift, and we do. When we moved them down the mountain, transplanted individuals spent far more time out of the sun, so they spent less time basking than the high elevation conspecifics. And we were interested in the costs associated with plasticity itself and with acute temperature changes. So we looked at body condition as a proxy for this cost. That is just essentially how much fat they're able to store, that kind of thing. And we looked at these different measures of how energy may be affected, both in terms of thermal condition and in terms of plastic, cost of plasticity. So we looked at energetic expenditure using doubly labeled water that's using stable isotopes to estimate the approximate turnover, and you can use that to do daily energetic expenditure. We also looked at corticosterone because it can liberate glucose and cause a more rapid decline in body condition, and it's also related to stress and has these weird interactions with temperature. And then finally we looked at spatial ecology to understand what might be affecting, how these animals might be acting different after this transplant. And that's partially to understand whether or not our transplant itself is affecting them, but also to understand if they're having to utilize a warmer environment differently. Because one thing that you can imagine is that as these shifts occur, we're gonna see shifts in plant communities and things associated with spatial use. So what we see first is that being moved down the mountain is costly. So this acute temperature does come at a cost. They lose substantially more weight when moved down the mountain than they do when they're kept higher up. So it seems that plasticity buffers the direct effects. You know, there's not this, I'm jumping ahead a little, sorry. So on this left-hand side, I've got my daily energetic expenditure. And while we would hypothesize that if they are experiencing these warmer temperatures and they're not able to get away from them, they would have this higher maintenance load. So they would be at a higher temperature, and that would cause a more rapid decline in body condition. We actually see the opposite. So the transplanted lizards used less energy daily than the control lizards. Then in court, we don't see a significant difference between the two groups in how they respond, though you know there is a little bit of a shift. It doesn't seem like it would be enough to explain why they're losing so much weight. So what this is telling us is that plasticity does seem to buffer the direct effects of temperature change, right? They don't seem to be experiencing this huge energetic load that we might have predicted, but it might shed light on some of the other costs that are associated with it. That is, they may be spending less energy because they can't be out and about as often. Because it's warmer, habitats have become unsuitable and they're having to spend more time in shelter. And another way that we can look at this is the space use aspect. And we actually see that while these aren't significantly different, there's a fair bit of variation. There's this qualitative difference in how they're using the space. that is these transplanted lizards are roaming a lot more, and that might actually be an indication that they're having trouble finding food and may explain some of that cost. So again, the cost of plasticity may be felt elsewhere. So, like I said, behavioral plasticity seems to be sufficient to buffer immediate thermal effects, but behavioral plasticity is costly. So this could come from a reduction in foraging time, but it also could be some experimental issue that we have not been able to parse out as of yet. Importantly, this mass loss could have really serious downstream consequences, right? If you don't have enough energy to keep your own weight up, to store weight, you're not going to have energy to reproduce. So what this can lead to is demographic collapse, where we're actually missing generations, especially if we have a heat wave at the right time of year, or we have a sustained heat wave. So again, this plasticity seems like it may actually be detrimental in constraining evolutionary response, because if these animals are plastic, but then they're not able to reproduce, they're not contributing to the next generation. They're not passing on any of these useful potential phenotypes. So with that, I'm going to spend just a minute or so talking about my current research, and then end with some projects I'd really like to do here. So the first thing is I work with these lizards called brown anoles. They're super common. They were introduced to Florida back in the early 1900s, and since then they've spread across the entire state, as well as many other areas. And these are really great system for a bunch of different reasons, one of which South Florida has this interesting, essentially our entire reptile community is non-native. So we have this completely novel community. So we can ask questions about how novel communities interact with one another. And this is actually a project I did as soon as I got to my postdoc. I worked with the data that these students collected in a herpetology class. So this is what we call a course-based undergraduate research experience. And I worked with them to write this paper up, looking at these behavioral interactions. And this actually just came out last week. The thing that I came to Florida to work on is this, essentially this really, really nice environmental climb. So that question of how physiology varies across the environmental climb can pretty easily be addressed in these lizards that we know a ton about, like when they were introduced at different points and whatnot. And Florida has this really great actual environmental climb that we've quantified, and we see that we see this increasing seasonality as we move north, and this decreasing temperature. So we can ask these really interesting questions. So the thing that I've started working on now is looking at immune function, the thermal performance of immune function, so quantifying it across thermal gradient, as well as looking at those upper and lower thermal tolerance limits, and the response of thermal plasticity itself. So whether or not these animals can rebound after being exposed to high temperatures, which we would also hypothesize would vary across an environmental climate. And then finally, I have this system that we've got everything in place to start up, but we haven't gotten to do it yet. And this is cool because because I came down to do this system, this is my system that would potentially be able to come with me. My advisor's been very gracious, and he's like, yeah, this is your thing. If you want to keep working on it, I'm happy to help. If you don't work on it, I'm probably not going to pursue it. So this is really cool because all these spoil islands have lizards on them already, and we have this really broad authority to manipulate these islands in any way we want. So we could remove trees, we can add cover, and see how that affects the phenotypes in real time, as well as look at things like parentage and understand how these traits may pass and how phenotypic plasticity may contribute to all that. So really kind of integrated into the context of my desert work, but with potentially a more known system and a potentially better experimental system that we can handle more closely. So then I'll just end with the things that I really would like to pursue if I were to come back here and get this job. And The snake system is really near and dear to my heart. Snakes were these nasty, disgusting snakes were my first love somehow. And then also some cool stuff that we could potentially do with some really local systems within Little Rock that would be really great for including undergraduates in doing some of the course-based undergraduate research stuff. So the first thing, like I mentioned, I'm really interested in the actual machinery, the mechanisms that are allowing this change in growth rate. So we know that consumption is the same, but we still see this higher growth rate. And one way of getting at that might be using something like transcriptomics, where we sacrifice animals at various states and we look to see what genes are differentially expressed and see if we can identify groups that are related to alternate pathways in growth or energy usage. So that is one that would be really great to work with. The transcriptomics is stuff that I'm familiar with and that I've done some extractions, know how to work with, but I have a great collaborator who has his lab set up for this. So this is something that I could really do and hit the ground running. And it's nice, systems like this would be brought into the lab. So there are things that, one of the constraints on some of the field biology is the animals just become inactive, but that's not the case if you're bringing them in the lab and growing them in common garden. Another question I would be interested in working on is how much more we can parse the relative contributions of adaptation and plasticity. The easiest ways to generally address adaptation and plasticity in a field context are usually common garden or reciprocal transplant type things. That is completely not feasible with these animals. They're too big, they move too much. Originally, when we were working on those fish farms, there was this goal, this lofty goal of doing a mark recapture study. And we had, after years and years of working on this study, we had so few mark recaptures, it was impossible to do anything with, just because there are so many snakes on these farms. But another way of getting at that is using quantitative genetics. So that's taking known parentage and assigning variants. So using these statistical models and methods to understand the relative contributions of the environment versus the genetics. So how much do the parents contribute to this offspring versus how much does the environment, based on how similar litters are to one another and different they are from one another. And this is one that I have some preliminary data for. These populations are really great for this. It is a daunting task because, like I said, some of these really large phenomes can have upwards of like 80 offspring. So you do have to go through and measure everything, but I think it's a really worthwhile project and something that's totally feasible within the context of the facilities that we have here. And then I just wanted to touch on some of the course-based undergraduate research experience stuff that I was mentioning. So there's this really great population of salamanders in the city that when they're breeding, they make this massive migration down from the hill to these ephemeral ponds. But because this is a city and where there's infrastructure here, they have this one big blockage, which is a railroad track. So they can get past it and they do this and they breed, they're just fine. But if you go out when they're breeding, you can find dozens and dozens of these salamanders along the railroad track. So we have this really great way of addressing some really interesting questions at the basic ecology level. So things like phenological changes in reproductive timing. But we can also, use these animals and look at things like salamander egg physiology. So this is a potentially really important question in a climate change context. These ephemeral pools are going to be less common with climate change. Timing of rain is going to change, stuff like that. But these are actually, so this is a spotted salamander, and they have this really weird system where this is not just muddy green water. This is an egg sac filled with algae because they have an obligate symbiosis with algae, and the algae provides the oxygen for the developing embryos. So there are a lot of potentially really interesting questions we can ask is there a way that we can inoculate these to where they don't have the algae and see how that affects it? Can we look at more ecological factors like how oxygenation of water affects the physiology of the eggs itself? So there's some really great questions that can be addressed within the confines of a lab. And as great as research is for those of us that go into the field, research isn't for everybody, but I think a research experience is valuable for all our students. So I'd be really excited to implement things like that. And with that, I've gone kind of long, so I will just throw out this acknowledgement slide and say thank you for having me, and I'm happy to attempt to answer any questions that you might have.
Great, thank you very much.
Thank you.
Okay, question. So let me start with the question. So when we talk about plasticity and adaptation, so you mentioned, let's say the precise effect the predator's size.
Yeah.
Is this reversible? So after you put it back from a big fish pond back to the small fish pond, is the plasticity back to the normal?
No, so this is actually looking at somatic growth. So this is actually the length of the animals. So they are,
okay.
yeah, this is not reversible. And while there are forms of, reversible plasticity that may be, so this was actually something that a previous student here was interested in, but looking at fat bodies, that might be something that where you'd expect, we talk about those changes in energetic machinery, you might see this shift towards storing a lot more energy that would reverse if they're moved to a small fish farm.
So if only it become longer, is it because of nutritional stuff, bigger fish, small fish? Or?
So that's one for sure effect, but it seems that because we kept consumption constant, that there's some shift in machinery to where they're differentially allocating the energy to somatic growth. That's not to say that that's always going to be the case, because those allocations, they shift rapidly and frequently throughout ontogeny. I think probably what happens is that these small female snakes, they're born bigger, so they already have that little advantage, whether that's from an adaptive response or a maternal investment. They are differentially, they're eating these larger predators, which is causing this differential growth rate to where instead of devoting a lot of energy to, say, maintenance or storage, it's...
But the model study is done in adopt. We do not talk about, development of those.
Right.
So it's, okay.
Yeah, so that's one of the real constraints with this species is because they get so big and it takes long enough. It's hard to have that intermediate, and particularly where we would see the biggest shifts in allocation would be around sexual maturity, and having that many animals in the lab is really tough to do.
Okay. So also you mentioned this mainly occurred in females. So basically this look like the only purpose for this adaptation is for reproduction.
I think that it's not the only purpose. I think it's a combination. So one thing, and I didn't talk, so this isn't a paper, but it kind of gets really muddy if you start trying to throw it in, talk about it. But males actually, we see these interesting shifts in their head shapes.
Okay.
It might be this idea of many-to-one mapping, where because males don't need to get bigger and it takes a lot more energy to be bigger, you might actually see a shift in their head size. Okay. And we do have some evidence of that, but that's another, that is because the head is so integrated and kinetic, identifying exactly which parts are important is difficult. But yeah, it seems like it might be adaptive, and they're just using different methods to go to the same thing.
For my curiosity, how do lizards increase the temperature?
How do lizards what?
Increase his temperature.
So lizards are really, particularly the lizards that I worked with in the desert, they're really effective thermoregulators. So when it's hot, when it's cold, early in the morning, is a really good time to go look for them because they'll just go to the biggest boulder they can find and sit on it to warm up. So they're, and not all lizards like that are like this, like tropical lizards. Because tropics are so stable, they typically they're where they are. But lizards here and in the desert, they are very active in shuttling around to find the habitat that's at the temperature that they want. And there's some studies that show that they are exceptionally effective at it, where you can compare their optimum temperature in the lab and then put them in a thermal gradient, and you can see that they are hitting that optimum temperature, and then you follow them in the field and you see that they are still throughout the day maintaining that same temperature throughout the day. So that's one of the things that's really important with the climate change, is that those methods that they've been using and the habitats that they've been using are gonna start to shift. Like, it's not even just the temperature, you're gonna see changes in plant communities. So, one of the things that we see in the high elevation desert is that with increasing elevation, you get more trees, which can be really helpful for thermoregulation. Where lower down, it's more like shrubby and grassland. So as temperature increases, there's this prediction that those communities are going to shift to the more grassland. So that's going to throw an even greater wrench in their ability to thermoregulate.
Another question for the snake and the fish story. So are there any possibility that some of your discoveries can be applicable to farming? So can you help farmers to increase the yield of fish or yield of a snake? Or?
Yeah, I think, and that's where, getting into the machinery that's involved in it, because, whether or not it's a consumption effect, it is still tough to, we're looking at this in a pretty small sample. It's possible there is this consumption effect that we weren't able to see. But yeah, if you can understand the machinery that is potentially causing these organisms to shift their allocation, you could theoretically turn that machinery on when you want it on. So, if you're like, well, all I'm doing is growing these fish to be as big as possible to go in somebody's pond, if you know how to switch that machinery on, you could potentially grow them much faster. And things like temperature are going to help with that too. But, understanding the underlying mechanisms is not just about this system. It can apply to other systems and potentially be applied to solving problems like that, beyond just the ecological, eco-evolutionary interests.
Hey, Maria.
How would you incorporate your research in your teaching here?
So I think the cures would be a lot of it. So obviously, my research is pretty conducive to experiments both in the field and in the lab, given the space and everything. But also, in ecology and say we're teaching the ecology evolution course, these things really they do go hand in hand. We tend to think of evolution, particularly, I know I did as an undergraduate coming in, this huge long time scale thing, but contemporary evolution is really important. And integrating my examples, but also other examples of folks from the field, and showing that evolution can matter within your lifetime on these time scales. Think of things like drug resistance and stuff like that. But I think the most obvious way that I would like to integrate it into my courses is developing research projects around, likely some physiology stuff, but also some basic ecology stuff like phenology and things like that. And Little Rock is really well suited to it because there are a lot of natural systems that we can go and explore. I know there are folks within the department, within the university that have access to these places, and as well as my contacts as well. And the salamanders at Murray Park, it's a public park. As long as you have the permits to collect the salamanders, nobody can tell you you can't collect the salamanders. So, yeah, I think trying to get some of this, the hands-on research and investigate questions beyond what we often see in ecology labs, how does this soil affect growth, is interesting, but you've probably done that in high school, so more direct understanding.
Okay. Any other questions? All right. Yes.
Like the growth rate and the like prey size and those features, are they affected by natural calamities other than the progressive climatic change?
Are they affected by what?
The natural sudden calamities like droughts.
Yeah, so the prey sizes are really constrained on those properties because they actually cull. So this pond that they, like, each pond has a known size fish in it. But the snakes themselves, you know, they're going to be affected through climate change. Like even early in the year, we can go out and find these snakes during the day. Right now, you would not see one of these snakes during the day. They're only out during at night, soaking up the last little bit of like, you can, if you've ever been to like a fish farm or even a lot of ponds have them, but the aerators, snakes love hanging out on aerators because they can get some of that last conductive heat. So I think there will be some other effects and we may expect like some changes in energetic quality of the prey items themselves with climate change. So you think if like the ponds are getting warmer, the prey items aren't necessarily processing their food as well. Processing faster, not pulling out all the nutrients they need. So that can affect the quality of the prey items as well.
So you know probably when at Five Lab we have very nice fish research and fish farms and stuff like this. So do you think you can collaborate with them, find way to maybe write grants together?
Yeah, I think so. It's something that I thought about a lot. I will say, I don't think that they realized how big this, like the farms that I worked on specifically, I don't think they realized how big the snake population was. You know, if you go during the day, you find dead snakes because when they see them, they kill them. But they definitely don't see very many of the snakes that are out there. So I think that there may be, I think there are ways that we can collaborate. It's just figuring out like what specifically we're interested in. I mean, one of those things might be like, what are effective solutions for these predators that you don't want on the farms? Having been on these farms for many years and caught thousands of snakes on them, you know, I'm sure that I can help in that kind of capacity as well. But yeah, I think UAPB would be a very good potential collaborator, especially if they have any interest in, like, they have this huge property with ponds. You know, if they have any interest in allowing us to experimentally manipulate, that would be great, because that is one of the biggest constraints. You know, like I obviously do a lot of transplant experiments. These animals we can't do that with because it's like those farms are too big, there are too many of them, but if we had the space and the right size ponds, we could do that kind of thing, and it could potentially be really interesting.
All right, great. So again, thank you very much for
Thank
coming to
you.
the seminar.
Yay!